Just check whether am I audible to you.
Yes, sir. Good morning, sir. Welcome.
Good morning, sir. Good morning.
Good morning and welcome.
Yes, sir. Can you hear me, sir?
Yes, sir. Clearly you are audible. There is no issue.
Okay, sir.
I will make you co-host. Now you are a co-host, sir.
Okay, okay. Thank you, sir.
Okay, sir. Good morning, Thackeray, sir. Dr. Thackeray. Sir, we'll wait for 5 to 10 minutes to participant.
Okay, sir. Okay, sir.
Then we'll start. Shall we start, sir?
Yes, sir. Yes, sir. You can.
Okay. Good morning to all of you and welcome Dr. Yogesh Bhagat, sir, for accepted our invitation and delivering lecture today. So, good morning to everyone. Dear participant, uh, today is the 8th day of this training program, and we have with us eminent personality, uh, Dr. Yogesh Bhagat sir. So I would like to happy to introduce our today's speaker, Dr. Yogesh Bhagat is currently working as assistant professor in molecular biology at VD College of Agriculture Biotechnology at Latur under the Vasantrao Naik Marathwada Krishi Vidyapit Parbhani. He has been recognized with several national awards like PR Verma Award for Young Scientist by Indian Society of Mycology and Plant Pathology, QAS Merit Award during post-graduation degree. He has also secured first rank in the state of Maharashtra for his undergraduate. So he has qualified 2 times for ARS interview during 2010 and 2015. So he has also secured the first rank for MCAR CET for PG admission in 2008. So he has been also nominated for various national fellowships like ICR, SRF and DBT JRF fellowship. So he also handled few projects include 2 DBT project, one ICR project, and presently handling one state government project of DNA fingerprinting of crop variety for public and private sector. He has also served in ICR where he licensed various technologies developed by the ICR and generated the revenue of around 30 lakhs during his tenure. So despite of this, his work has been presented in several publications, including 20 research articles, so various book and book chapter published in reputed peer-reviewed journal. Okay, so with this word, I kindly request to Dr. Yogesh. So please share your screen, sir, and deliver the lecture on the said topic. Please, sir.
Thank you, Bharat sir. Actually, we have preponed this lecture due to some unavoidable circumstances. So I'm thankful to Bharat sir for considering the early morning the time. I think is it fine for all the participants this time?
Hello,
Is
sir.
it okay?
Yes, sir. Yes, sir.
All of you are you ready there? Yes. So in previous lecture, what we have seen about the current scenario of genetically engineered plants at national and global level. So there, what we have seen that uh, in all over the world at a global level. So what is the area that has been occupied by this transgenic crop or genetically modified crops? We have also seen which is the first transgenic crop, which company has developed that transgenic crop, and for which trade that transgenic crop has been developed. Then which is the first cereal where the transgenic crop has been made, which is the first forestry where the transgenic crop has been made, which is the first flower plant where this genetic engineering technology has been implemented and commercialized. Then we have also seen which are the countries, which are the main growers of these genetically modified plants all over the world. So, which is the first, the country which ranked first, second, third, fourth. Then we have seen which crop is having the highest area of cultivation under this GM crop. That means which crop, okay. Then we have seen which trait, okay, whether it is the insect resistant, whether it is a herbicide tolerant, or whether it is a herbicide resistant trait, or disease resistant trait, or oil quality, or any quality trait. So which trait is dominating among the GM all over the world? Then we have seen the various transgenic crops which are in the pipeline for its commercialization in India. So there we have seen about the Bolgard 1, Bolgard 2, that is the BT cotton cultivars. Then we have also seen about the BT brinjal. We have seen about the status of transgenic crops in Asian countries. We have also seen the golden rice, which has been developed for the quality improvement. We also have seen the herbicide tolerant STBT cotton, that is Roundup Ready Flex. How it is developed, who have developed that, what has happened to that. Then I have introduced you about some committees that is which regulate this transgenic research or genetically modified plant research. So these committees that I have introduced you about that various organizations, various committees, they play an important role in the regulation of these transgenic plants. Then we have a little bit we have seen about the genome editing. Okay, so if there is some because you know that and there we have also seen the what about this GM byproduct or genetically modified plants byproduct which are in our food chain,
hello, sir.
like cotton seed oil.
Hello, sir.
Hi, yes.
Sir, please.
Yes.
The slide is not moving.
No. I'm giving the previous lectures background.
Okay, sir. Sir, and make
previous lecture so that
yes, sir. Please
there will
make
be continuation. There will be
horizontal.
link, okay. Oh, it's in this one land. What I say orientation. Just I will check it. So in the previous lecture, that means whatever the genetically modified or transgenic byproducts are there in our food chain, that we have seen. Then We have also seen the whatever the edible oil we are importing from foreign countries. So you know that around 400 million metric tons of edible oil we are importing from the foreign countries. So that edible oil is also have been made from that GM crops. So ultimately, it's in our food chain, and we have also seen that some of the vitivinicol seeds are coming from Bangladesh. Farmers are illegally cultivating the STBT seeds, so that is also there. So still the government has not took any decision for the whatever the next vision for this new technology. So that is genetic engineering technology or genetically modified plants. And we have also seen that this is the very much important technology when we talk about the revolution in the field of agriculture. So we have to adapt some such a newer technology. So today we are going to see a little bit background or breakthroughs in the field of molecular biology. You know, the molecular biology is one of the branch of that genetic engineering or biotechnology. Okay, so we will see about the little bit breakthroughs, which are the scientists which has played a major role when we talk about the biotechnology or molecular biology. Then we'll see what are the different methods by which we can make a transgenic plants, how the transgenic plants are being made. Then once transgenic plants are being made, so what is the how to confirm whether transgene is there, whether transgene is expressing, whether transgene is making a protein. Then we will also see what are the regulatory bodies and their role when we talk about the commercialization of transgenics. So just I will start it from the historical things, like what are the major breakthroughs, which scientists played a major role when we talk about this new technology or genetic engineering or molecular biology or biotechnology. So just look at this scientist contribution. These are the major breakthrough when we talk about this new technology. You will see the contribution of that various scientists in this field. So you know, in Darwin, in 1859, he has given the phenomenon of evolution. Okay, origin of species. So that origin of species. So in this he said that living things which are the result of natural selection. So during the course of evolution, so whatever the organisms are exist, so they are the result of natural selection. Okay, and in 1860, just 1 year after that, Darwin's idea supported by Gregor Mendel, you know, father of genetics, right? And he found that genes determine the characteristics a living things will take. So what actually Darwin's idea was there? So evolution. So Darwin said that whatever the organisms exist on the earth, they are having a common ancestor. So even birds, animals, human beings, insects. So all we are originated from a common ancestor. We are similar in terms of certain metabolism. We are similar in terms of some morphological characters. We are similar in terms of various biochemical parameters. So theory of evolution, or Darwin said that all living organisms have originated from common ancestor. And what Mendel has said that genes determine the characteristics. You know what Mendel has given? Heredity concept. Heredity means there are some characters which are getting transmitted from parent to offspring. Means there is an heredity. He has not said how they are getting transmitted, but he has said so there is an heredity. And whenever heredity and variation, so the characters that they are getting transmitted from parent to offspring, or one generation to the next generation. And what Darwin said, we are having a common ancestor. All whatever the living things are exist on the earth, we are having a common ancestor. So something has been inherited from that common ancestor, and there is a divergence and there is a variation during the course of natural selection. So uh, uh, the life, living things are exist on the earth. So what Mendel has given in 1866? So he has given the law of segregation, law of independent assortment, and law of dominance. So law of segregation, what it states, a gene pair defines each inherited trait and gets randomly separated to the sex cells, which ensures offspring to inherit one genetic allele from each parent. So you know that one allele, so in a diploid organism, so there are two alleles, we said that, and that each allele has been come from either male parent, one from male parent and one from female parent. So there are two alleles, okay, and they are located on the homologous chromosome. So each gene has at least one allele. So they are located on these homologous chromosomes, and that homologous chromosome means one chromosome is coming from male parent and one chromosome is coming from female parent, okay, and means one allele is coming from one parent and other allele is coming from male parent and other allele is coming from female parent. Then law of independent assortment. So gene from or different traits are sorted from one another in such a way that inheritance of one trait is not dependent on the inheritance of another. So genes are getting segregated randomly. Okay, genes are getting segregated randomly. We suppose there is a gene and there is B gene. So segregation, inheritance of A gene is not dependent on the inheritance of B gene, or inheritance of B gene is not dependent on the inheritance of A gene. Actually, this has been overcome by the law of linkage. Okay, that I will come later. Okay, but in 1866, what Mendel has proposed, that I am saying here. But that time, actually, this Mendel's law were not accepted. Okay, actually, Mendel's work has been carried forwarded by De Vries, Koresh, and Teichmann. And then, when they have independently worked on this Mendel's hypothesis, then finally they have come to know that. Then they have finally given the confirmation for this law of segregation and law of independent assortment. Then 1869. So 1869 research scientist who has given the term nuclei. Okay, so he has isolated the white blood cell. He has isolated the DNA from the nucleus of WBC. Means that time it was not known. It is it was a DNA. Okay, so whatever the things he has isolated from that nucleus of a WBC or nucleus of a white blood cell, so he has given that given the name for that substance as a nuclein. Okay, later, right now we call it as a DNA. Okay, and he has characterized that that substance has some elements like phosphorus, nitrogen, and sulfur. So, Mr. who has given the term nuclein for the whatever the substance he has found in the nucleus. Then Sutton and Bowry, they have given the chromosome theory of inheritance. Sutton and Bowry, they have given the chromosome theory of inheritance in 1902. Then Johnson in 1909, he has given the term genes, which are the unit of heredity. He has also introduced the term genotype, that is the genetic constitution of an organism. Phenotype, as an organism totally inherited characteristics. So what phenotype it will get reflected, what are the morphology that organisms display. So it will be because of genotype. So the scientist Johnson, he has introduced the term gene, genotype, and phenotype. Then in 1910, so Morgan, so Thomas Morgan places a gene on chromosome and discovers the phenomenon of crossing over, and linkage. So in law of independent assortment, which has been given by the scientist Mendel, so there I have said that A gene is independently inherited with the independently inherited as the B gene, means A and B, C, D genes. These genes are independently inherited, means their inheritance is not dependent on one another. But there is an exception for that. So Morgan has discovered the phenomenon of linkage. Linkage means if two genes are very close together, if their genetic distance is less than 50 centimorgan, then we say that two genes are linked together. Means suppose A and B genes, if their genetic distance is less than 50 centimorgan, so genetic distance we measure in terms of centimorgan. Whenever we say that two genes are having a genetic distance less than 50 centimorgan, means we say that genes are linked. Means they together get inherited. Suppose a distance between A and B gene is less than 50 centimorgan. So there is a chance that the genes A and B genes are getting inherited together in a progeny, in a offspring, from one individual to other individual, from one generation to the next generation. And if that gene distance is less than 20 centimorgan, then we can say that there is a tight linkage. Then we can say that there is a tight linkage. So the student of Morgan named Alfred Stutevant, who has created the first genetic map of fruit fly genes. So genetic map means we are fixing the position of genes on a genome or on a chromosome. So where that gene is located on a chromosome, where the gene is located in a genome. Okay. So he has mapped that location of a fruit fly gene. So mapping means what? So like we can find out the position of a gene on a chromosome. We can find out the distance between different genes in a chromosome. Okay, we can find out the distance between. Just mute mic. Mic is on. Chetan Shinde, just put your mic on mute mode so that there will be no disturbance. So first genetic map has been created for the fruit fly genes, means the location of genes, the gene distance, everything has been mapped in fruit fly. And the student of Morgan, so Stutevant, who was the student of scientist Morgan, has given the first genetic map for the fruit fly genes. Then in 1928, you know that Fred Griffith experiment. So what he has done. Chetan Shinde, just I will mute you from there. So Griffith, what he has done in 1928. So you know that rats or mice is has infected with some pneumococcus bacteria. Okay, so once there is an infection of that pneumococcus bacteria to the mice, so mice will get died. Actually, there are two strains of that pneumococcus bacteria. So one is S strain and one is R strain. So S strain actually infects the bacteria. Sorry, it actually infects the mice and mice get killed. Yes, smooth strain. And in rough strain, when rough strain, there is an infection of that rough strain to the mice. So mice is not getting killed. So what he has done, so where yes strain is there, which is actually when it infects and mice is getting killed, what he has done, sorry. Actually slides. So what he has done, as I told you, there were two strains of a pneumococcus bacterium. Okay, and what he has done, so in pneumococcus, yes strain and R strain. Yes strain, whenever a smooth strain, whenever there is infection with smooth strain, the mice was getting died, and whenever was an infection of R strain, the mice is getting survived. So what Griffith has done, he has killed that smooth strain, and he has mixed that smooth strain culture with yes strain culture. But when he has mixed, and when he has infected that mixture with the mice to the mice, then again mice is getting died. So even though he has killed that yes strain, which was infecting virulent strain, that is a smooth strain of pneumococcus, he has killed that, and he has mixed with the rough strain. Then also mice is getting killed. This he was found. He was hypothesized that there is something which is getting transmitted from that smooth strain to the rough strain, and that confers that mice get died. Okay, so he has given the term that is the transforming principle. Now, every McCarthy beyond in 1954, So what they have seen, so whatever the heat treated material was there of that smooth strain bacterium or virulent bacterium, this Ivory, McCarthy and McLeod, they have used the various enzymes to treat this mixture. So like proteases are there, ribonucleases are there, RNAs are there. RNAs is which degrade the RNA, proteases which degrade the protein, DNA which degrade the DNA. So right, these 3 to 4 reactions they have carried. Like first they have treated that mixture with the proteases, but they found that okay, whenever they treating this mixture, that S strain and S R strain, then also moist spice was getting died. Then they have used the RNA, ribonucleases, RNAs. Means RNAs will degrade the RNA, so then also mice is getting died. When they have used DNAs, means which degrades enzyme which degrades the DNA, that time mice was survived. So what they come to know, so if the DNA is actually the responsible for this transforming principle, means DNA is getting transmitted from that uh, smooth strain, virulent strain to the avirulent strain, and that's why whenever we injecting this such a mixture to the mice, that's why the mice is getting died. So DNA was the transforming principle. That time Andy McLeod and McCarthy came to know. Then McClintock, she discovered the genetic elements, that is the mobile genetic element. So means there are some genetic gene segments in a genome which moves themselves, which moves from one location to the other location, and that's why they create a variation. So they, McClintock, she studied that phenomenon of transposition or transposable genetic element or mobile genetic element in maize. And in 1983, she was also awarded with the Nobel Prize for discovery of transposable element. Then Charga, they have given in 1950. So he said that there is a regulatory in proportion of DNA basis for different species. So purine is equal to pyrimidine. Okay, so A plus G is equal to P plus G. So this ratio is constant. So whenever we say that purine, you know, adenine and guanine, they are purine, thymine and cytosine, they are pyrimidine. So there is an equal proportion of these purine and pyrimidine. Then Hershey and Chase, they identified DNA as a genetic material of harvest. You know, bacteriophage. So bacteriophage, it infects the bacteria, and that bacteriophage is viruses, they are made up of DNA and protein. Viruses are made up of DNA and protein. So what they done, they label the DNA separately, and they label the DNA with phosphorus, and they have labeled the protein with sulfur. Okay, radio label. And what they have found, when the viruses or bacteriophage infect the bacteria, that time that bacteriophage incorporate the DNA inside the bacterium, and after that there is an agitation, it will get separated. Okay, so they have found that, so once they have isolated that bacterial suspension, they have identified that there was no protein inside the bacterium, but it was the DNA which was radio labeled with the phosphorus P32. Okay, so whatever the sulfur label protein was there, that they have found in supernatant. So whatever the bacterial palette they have found, so inside the bacterium, only DNA content was there. So in such a way that they have come to know. So this is the DNA as a genetic material which this bacteria are incorporated inside the bacterium. Okay, so that time viruses were known as only the DNA was the genetic material in viruses, but Conrad and Singer, they have also shown that RNA as a genetic material in the viruses. Not only DNA, but RNA also acts as a genetic material in some viruses. So majority of the plant viruses, they contain RNA as a genetic material. Then Watson and Crick. So in 1953, we all know that in 1962, Watson and Crick, they have given the double helical structure of DNA. Okay, and they also awarded with Nobel Prize in 1962. So you know that DNA is double-stranded helical structure. There is a minor groove, major groove. There is equal proportion of purines and pyrimidines. So distance between each nucleotide is 3.4 degree angstrom. Distance between 10 nucleotides, it is 34 degree angstrom. Diameter of DNA is 20 degree angstrom. So all these things they have given in their double helical structure. And that double helical structure has been studied by the technology X-ray crystallography. Then Kornberg, actually, they have discovered these enzymes, RNA and DNA polymerase. Okay, they have discovered the enzyme RNA and DNA polymerase, and they have been also awarded with the Nobel Prize in 1959. So these are the major breakthroughs in the science. These are the major breakthroughs in the biotechnology or molecular biology. So Crick has proposed a central dogma. Central dogma means it is DNA is getting transcribed into mRNA, mRNA is getting translated into a protein, and DNA is getting replicated to the DNA by the process of replication. So this was the central dogma which has given by the scientist Crick. Then Michelson and Stahl, so they have said that there is a semi-conservative mode of replication of DNA. Means the new strand of DNA is getting synthesized from their parental strand. Then in 1961, Jacob and Monod has given the Operon concept. So operons means in a bacteria or in a prokaryote, group of genes are under the same regulatory region. Means there is a group of genes which under the control of same regulatory region. But in eukaryotes, it doesn't happen. So all genes have their independent regulatory things. But in case of prokaryote, the group of genes which are under the same regulatory system, so we call this concept as a operon. Then Nirenberg. So Nirenberg, who has given the genetic code. Okay, you know that genetic code. Genetic code means you know the ATG C base pairs are there, and that 3 nucleotide triplet we call it as a genetic code. We suppose ATG is there, so ATG, and when it gets transcription, so it will synthesize AUG. So AUG means it is a in the mRNA, and AUG once it gets translated, so it will synthesize a one amino acid, that is glutamine. So code, codon, and finally amino acid. So genetic code concept has been given by Nirenberg. So there are 20 amino acids, and which all are these 20 amino acids are getting synthesized from 61 codons. And there are actually total 64 codons. Out of these, 3 are stop codons, 1 is start codon, and in total, if you see, the 61 actually codes for a 20 amino acids, because out of 64 codons, 61 3 codons are stop codons. So Hargobind Singh Khurana, who has discovered this DNA ligase enzyme, and he has also synthesized the oligonucleotides. So artificial synthesis of a DNA. And Khurana was also awarded with the Nobel Prize in 1968. Then major breakthrough in the gene cloning or recombinant DNA technology. That is after the discovery of restriction enzyme. So it is 1968. So the discovery of restriction enzyme was carried out by Urban, Smith, and Nathan. Okay. So they have discovered the restriction enzyme. So these are the enzymes which cuts the DNA. These are the enzymes which cuts the DNA. Then Tabin, Delbaco, and Baltimore. So Tabin, they have discovered the reverse transcriptase. Now, if you see the central dogma, means DNA is getting transcription into mRNA, and mRNA is getting translated into a protein. So from mRNA, there can be synthesis of the DNA by the enzyme reverse transcriptase. So they have discovered this reverse transcriptase enzyme in some viruses. And Tabin and Baltimore, they have discovered the reverse transcriptase enzyme. Means DNA is getting transcribed into mRNA, and mRNA also can reverse transcribe into DNA. So from our mRNA, there can be synthesis of the DNA. We call it as a complementary DNA or cDNA. So that is a central dogma. Then Kornberg. So Kornberg has given the concept of nucleosome. That is the basic unit of a chromatin in 1970. He also received the Nobel Prize. Then Biddle and Tatum. Biddle and Tatum has given the hypothesis in 1970 that is one gene synthesizes one enzyme. But recently it is modified as one gene synthesizes one polypeptide. Because there are some proteins which are made up of, there are some enzymes which are made up of more than one polypeptide. So these all polypeptides are getting synthesized by individual genes. So initially, Biddle and Tatham's hypothesis was one gene, one enzyme, but now it has been modified as one gene, one polypeptide. Then Paul Berg, who was the scientist who created first recombinant DNA. Paul Berg scientist created first recombinant DNA. So the human insulin. So you know that the insulin, whatever the insulin used for the treating the diabetic patient, or whatever the interferons which are being used for treating the cancer patient. So they are the products of biotechnology. These are the products of recombinant DNA. These are the products of genetic engineering. And most commonly, you are familiar about this corona vaccine, okay, which we have taken. So they are the recombinant vaccines. They are the product of genetic engineering. They are the product of this molecular biology. Then Stanley and Boyer. So they have given the birth for genetic engineering, that is DNA cloning. Sanger, who has done the DNA sequencing. So the first sequence genome was FAS174. Okay, first sequence genome was FAS174. That was virus. Okay, and he was his second Nobel Prize. He has his second Nobel Prize in 1980. Then Milston and Kohler has developed a monoclonal antibody, and they have also been awarded with the Nobel Prize in 1984. Okay, so these are the major scientists. So Carey Mullis, who has discovered the PCR, that is the polymerase chain reaction. So these machines can develop a large, that means you can form a millions of copies of that DNA with this PCR machine, and it has been discovered by Carey Mullis. So he has used the thermocyclical DNA polymerase in PCR. Okay, and he has also awarded with the Nobel Prize in Chemistry in 1993. So you can see here, the most of the scientists are associated with the molecular biology or genetic engineering. They are the Nobel Prize winners. So you can see the importance of this new technology. So Flavor Saver, that I have told you in the previous lecture, that was the first genetically engineered plant which has been developed by the Calgene company, and it has been approved by the US in 1996, and it was the Flavor Saver tomato. It was having the property of delayed ripening. The technology it used is the antisense technology here. And whatever I said, these transgenic crops are under the, so whatever till now we have seen about the major breakthroughs of this related to this biotechnology, related to this molecular biology. So we should remember the contribution of the scientists, because as you know, as they are awarded with the Nobel Prize, so you can see that whatever the discovery they have made, so you can see the importance of their discovery, whatever the discovery they have made. So we can right now you can taking, these are the workful discoveries, workful technologies, whatever they have discovered. So transgenic plants, that in previous lecture I have explained so much for this. So you know that if you have to work on the transgenic plants or genetically modified plants, so first you have to do the application to the institutional biosafety committee. Okay, so that institutional biosafety committee will approve your work related to recombinant DNA technology or genetic engineering. Then there is a regional review committee on genetic manipulation, regional committee on genetic manipulation for the greenhouse trials, or for BRL1 trial, that is biosafety research trials, which are conducted at some confined location in greenhouse or in less than 1 acre of area. Okay. Then there is a genetic engineer appraisal committee. So this is the apex committee event for the approval of this genetically engineered plant. So that is genetic engineer appraisal committee, and they conduct the various trials like BRL2, means biosafety research level trial 2. Means they carry out this multi-location trial at larger level, larger scale, is more than 1 acre of area. Okay. Then so this there is a someone certification committee, so which ensures that everything is getting in the proper regulations or as per the rules and regulations of that transgenic crops. So that there is a CCC, that is a certification committee which monitors that is which was in along with this RCGM and genetic engineering project committee. And ultimately, once GEAC gives that final approval, then the Ministry of Environment and Forest will approve that particular plant for the commercialization. So now we have seen about the transgenic plants and their status, global status. We have seen about the major breakthroughs and in the field of this molecular biology, various Nobel Award winners. Can we see here how the genes look like? What is the structure of gene? Okay, so I told you that there is central dogma means DNA is getting transcribed into a RNA, RNA is getting translated into a protein, and ultimately that protein is resembling a particular phenotype. So that RNA is whatever the RNA is getting synthesized is getting reverse transcribed into DNA. So you know that there was a discovery of that reverse transcriptase enzyme afterwards. Means RNA is also getting reverse transcribed into DNA, and we call that DNA as a complementary DNA or cDNA. So central dogma has been modified. So this is the recent central dogma whatever we have seen. And at the left side you can see there is a gene structure. Okay, at the left side that there we can see that there is a gene structure. So gene structure means it has promoter element, it has open reading frame with start codon, stop codon, and there are some regulatory sequences. This is the eukaryotic gene structure where you can find the intron and exon. Okay, so promoter is the site where this RNA polymerase binds, which synthesizes the RNA. Means it is a DNA dependent RNA polymerase which synthesizes the RNA by using DNA as a template. And this promoter means it is a nucleotide sequence only, but this promoter is the site where this RNA polymerase enzyme binds and it starts the transcription of DNA or transcription of gene. And this, in this way, the mRNA is getting synthesized, and mRNA is getting synthesized with this open reading frame. It means up to start codon to start codon, stop codon. So whatever the region between start codon to stop codon, we call it as an open reading frame. Once the mRNA is getting formed, this mRNA is getting translated, and there is a mature mRNA is getting formed, that is with 5' gap and 3' poly A tail, and that protein is getting then functional protein. Okay. So, what are the techniques of gene transfer into plant? We have seen in general the what structure of gene. So, whenever we have to insert a particular gene into a plant or any animals, whenever we think about the transgenic plants or GM plants, so this is the tDNA construct. These are the where it has the marker gene, where it has the promoter, where it has the transgene. So can we see there in blue color there is a transgene. Near the transgene there is a promoter element, and near the promoter there is a marker gene. So transgene, you know, which the gene which we are going, which we would like to transfer into a particular host. It may be insect resistant gene, it may be disease resistant gene, or herbicide tolerant gene. So it is under the control of promoter. So we have to develop all cassette. Okay, so this promoter are the elements where that RNA polymerase binds. So that gene, as I have said, the eukaryotic gene has their independent regulatory system that is like promoter. So it has its own promoter. Okay, so promoter are the site where the RNA polymerase binds. Then marker gene. Okay, then marker gene. So marker genes means these are the antibiotic resistant genes. So whenever we transform a particular gene, this cassette, we call it as a transgene cassette. Okay, it has the transgene, it has the promoter, it has the marker gene, right? So marker gene means they are nothing but the antibiotic resistant gene. So whenever we transfer this entire cassette into a plant, so we allow that tissue to grow an antibiotic. So the tissues or cells which carry only this transgene cassette, which will only grow on that antibiotic medium because of that marker gene. Because they are having antibiotic resistant gene, they degrade that antibiotic. So cells which carry that transgene cassette or cells which carry that particular gene of interest, which only grow on antibiotic containing medium, and we call that antibiotic as a selectable marker gene. Okay, selectable marker gene. So these marker genes help us to select the cells which carry this transgene cassette or the cells which carry the gene of interest. So there are various methods of gene transfer into a plant. That is physical method, it is a chemical method, it is a biological method. So both physical and chemical method in together we call them as a vectorless method, or we call them as a direct gene transfer methods. Okay. So indirect gene transfer methods or vector mediated gene transfer methods, we also call them as a biological method. So here we are using some bacteria like agrobacteria. But in case of physical and chemical methods, we are not using bacterial like agrobacterial. So these are the direct gene transfer methods, physical and chemical. Biological, there we can use some bacterial like agrobacterial. So gene transfer techniques in plant, as I said, direct gene transfer is there. So this direct gene transfer again is classified as a physical and chemical methods. Okay, so physical contains the gene gun method. This direct gene gun method, direct incorporation of directly we can input the transgene into a host or cell or tissue. Electroporation, microinjection, they are the physical methods of direct gene transfer. Chemical method means liposome mediated method, polyethylene glycol mediated method. And there is one more method that is an indirect method, or we call it as a vector mediated gene transfer, where we are using the agrobacterium to mediated gene transfer. Generally, an indirect or vector mediated gene transfer method is most commonly used method of gene transfer. Okay, agrobacterium purification is most commonly used method of gene transfer. You should remember that. So there are different methods of gene transfer: direct, indirect. Direct method are also called as vector gene transfer. Indirect method are also called as vector mediated gene transfer because we are using a bacterium for that transmission of a gene, transgene. In the direct gene transfer, again, it is classified as a physical, chemical. So physical means particle gun, electroporation, microinjection. Then chemical method, that is a liposome or pH medium. So in particle gun method, what we are doing? So DNA is coated with the DNA. Okay, so our gene of interest. So that gold particles or tungsten particles are coated with the gene of interest or transgene. Okay, gold particles or tungsten particles are coated with the gene of interest, and they are loaded in the gene gun. And there is a we can at higher pressure that gold particles or tungsten particles which are coated with the gene of interest are fired on the various tissues, and that tissues are allowed to grow on a plant tissue culture medium. Okay, so the tissues which are having that transgene cassette along with the selectable marker genes, which only grow on that particular plant tissue culture medium, and in this way we can generate a transgene plant. Okay, so this is the gene gun method where we are coating the gold particles or tungsten particles with gene of interest. Then electroporation. So here we are subjecting, here we need a chloroplast. So you know that cell without cell wall, we can't call it as a chloroplast. So first we need a chloroplast there. Then we are taking a DNA and we are allowing that DNA mixture of DNA and chloroplast together. And we are subjecting that DNA and chloroplast mixture or transgene and chloroplast mixture to a under a electric field. So that because of that pulse or electric field, so there will be some formation of some pores on the that plasma membrane. And during this formation of pores, that transgene is getting inside the that protoplast. Okay, and when we stop that electric field, then again that protoplast or that plasma membrane will regain its structure and it will form a cell membrane there. Okay, in such a way that that particular transgene is getting incorporated into a particular tissue or cell. So this is the method of electroporation where we are applying the electric field. Here we need a protoplast. Okay, and that also we are going to cultivate on a tissue culture media. And there we have seen gene gun method, which is also one of the method of gene transfer. Then microinjection. So here we need an electron microscope kind of thing because just a minute. Who is this? So microinjection, it is also a method of direct gene transfer or vector-led gene transfer, where we need a microscope, where we can, you know, that electron microscope, we can clearly see the nucleus of a cell, and directly we can inject that DNA inside the nucleus. Okay, by using various microscope. Here protoplast is needed, and this is marker free method. Okay, for the transgenic development, that is microinjection. Then liposome mediated. So liposomes are these are the lipid molecules, and that lipid molecules are having aqueous layer surrounded on that liposomes. And when we mix that liposomes with the DNA, so there liposomes adheres to the cell. Okay, and then DNA enters from that liposome adheres to the cell to the nucleus of the cell. Okay, so this is the liposome mediated method. which is a chemical method. We can call it as a chemical method of direct gene transfer. So next is indirect method of gene transfer or vector mediated gene transfer. As I told you, here we are using the agrometric gene transfer. So these are the indirect method of gene transfer or vector mediated transfer. This is one of the soil bacterium, so which forms a tumors or bubbles to the when it infects a plant. Okay, and it can be abundant in the soil. So what we had to do? So agrobacterium 2 patients, it has a TI plasmid. So in this TI plasmid, we have to isolate that TI plasmid. The TI plasmid has to be cut with the restriction enzyme, and then gene of interest has to be inserted to this TI plasmid to form a recombinant plasmid. Means the TI plasmid, which is present in the agrobacterium 2 patients, so that has been modified by restriction enzyme. We are making the cut to the this TI plasmid, and then orange DNA, the gene of interest, which gene we would like to transform. So that gene has to be incorporated into that particular TI plasmid. And in such a way that recombinant plasmid is getting formed, and that recombinant plasmid again we have to transform into a agrobacterium 2 mutation by the process of transformation.
Sir,
and that recombinant, yes sir.
Sir, please keep your mobile phone horizontal.
Sure. Shall I? Okay. Okay. So it will be just. Shall I keep it orientation like this? Is it moving now? Can you see? Is it moving, sir?
No, sir. Okay. Continue, sir. Continue. No issue.
Shall I continue?
Yes, sir. Yes, sir. Please.
Oh, has it changed? Has it changed its orientation or no? So the agrobacterium incubation, as I told you, whenever we talk about the gene transfer technology, agrobacterium mediated transformation method is most commonly used method of gene transformation. And here we can see we have to form a recombinant agrobacterium, and that recombinant agrobacterium which we have to allow, which are co-cultivated with the plant cell, the cell which would like to transform, the cell which would like to transform a transgene. So once we co-cultivate that agrobacterium, recombinant agrobacterium, which carries that recombinant TI plasmid or which carries that transgene or gene of that we have co-cultivate in that cell culture. That plant cell, once we co-cultivate that agrobacterium with the plant cell, so that agrobacterium has we transfer that tDNA into a plant cell. Okay, and then that plant cells we have to allow it to grow on some tissue culture medium. And this plant, again, I have said that transgene can have stable marker genes, means the genes which confers the antibiotic resistance, and that allows that particular plant to grow. Okay, so the cell does not have that transgenic acid. The cells which do not have that selectable marker genes like ampicillin resistant gene, kanamycin resistant gene, which are not being survive on that antibiotic containing medium. So that is one of the steps where we can select only the transform cells, and that only cells which allow to grow, and then which will form the entire plant by the process of culture. So next, so we have seen the direct method, we have seen the indirect method of gene transfer to the plants. Now the most important thing is the how to confirm this plant, how to confirm the transgenic status of this plant. Okay, how to confirm the transgenic status of this plant? Can anyone read what are the different methods for the confirmation of transgene? Any one of you can unmute. I will ask somebody to unmute. Okay, I am seeing so many are there. Okay. I will ask somebody to read it. Okay, Harish, can you read it? You just unmute yourself and you read it. What are the different methods for the confirmation of transgene or transgenic plants? I think he's not there. Uh, Monica Andre, can you read it? Can you unmute yourself? Just unmute yourself and read what are the different methods for the confirmation of transgenic plants. So, anyone would like to see that? Anyone like to read that? See, the first method of the most commonly used method of this transgenic confirmation is by the PCR based method. Okay, so the PCR, as you know, the PCR technology has been PCR. Just a minute. So this PCR technology has been invented by the Caribbean scientists. There is a other method of confirmation of transgene, that is southern hybridization. So northern hybridization and RT-PCR or real-time PCR or reverse transcriptase PCR, these are there we can check whether genes is expressing or not. PCR and southern hybridization, there we can know that whether that particular transgenic plant having the transgene or not. Northern hybridization and RT-PCR, there we can confirm whether that gene is transcribed into mRNA or not, where that mRNA of that particular gene is present is there or not. So expression we can study there, northern hybridization and RT-PCR. Then Western blotting. So Western blotting is also the confirmation of whether that gene is expressing or not. So whether that gene is producing a protein, whether that gene is getting mRNA is getting translated into a protein or not. So protein confirmation can be done by Western blotting. mRNA confirmation can be done by RT-PCR and northern hybridization. Means in total, expression of that gene can be confirmed by northern hybridization, RT-PCR and Western blotting. And how to know the presence of transgene? So it is by the method of PCR and southern hybridization. So the transformation technique, okay? So transformation technique. So whenever we do the transformation, as I said you, that transgene cassette. So that transgene cassette it has these selectable marker genes. Okay, that transgene cassette it has a selectable marker genes, and that selectable marker genes allow or help us to select the cells which carry that transgene. Okay, so antibiotic, we are growing that particular cells on the antibiotic containing medium, and we are allowed. So whenever we do the transgenic, whenever we produce the transgenic plant, so there will be chance that whatever the plants are being generated, whether they carry so whenever we generate the transgenic plants, so there is a chance whether that plant will have the transgene or not. So there will be some escape. Again, there will be question if there is a presence of transgene, whether it is transcribing, whether it is expressing or not. Again, there is a question whether it is forming a protein or not, whether it is form a protein, whether it is form a RNA, whether there is a presence of transgene. So all these possibilities can be checked by this transgene confirmation. So as I said, PCR and southern hybridization, these are the methods to check the presence of transgene. Northern hybridization, RT-PCR, real-time PCR, these are the methods to check their RNA, whether that transgene RNA is there or not. Western blotting, Western hybridization, these are the methods to check the protein. ELISA is also to check the protein, whether the protein synthesized from the gene is there or not. So polymerase chain reaction. So in polymerase chain reaction, what we are doing in polymerase chain reaction? So in polymerase chain reaction, it is a confirmation of presence of transgene. So this technique is used to confirm the presence of transgene. Okay. So here what we are doing, then we are using some primers to where that PCR polymerase chain reaction. Okay, here we are using some primers which are specific to their transgene, okay, which are specific to the transgene. And when we are producing a transgenic plant, so total genomic DNA from that transgenic plant we are isolating, and we are subjecting that total genomic DNA of that transgenic plant for the PCR with the primers, which are the complementary sequence to the transgene. So that particular gene is getting amplified in the PCR. That gene is getting produces and millions synthesized in the millions copies in the PCR machine. Whenever we are using the primer, that is the complementary DNA sequence to the transgene. Okay, so we are running. We can find out the. So here we can see the black color image is there, and we can see that there are some bands. So these are the DNA bands. So like 400 base pair, 500 base pair bands are there, which indicates that there is a presence of transgene. Okay. So polymerase chain reaction. So that confirmation we can do in the various generation of the. So in breeding there is a F1 generation, F2 generation, F3 generation. So like that in transgenic also there is a T0 generation, T1 generation, and T2 generation. So actually T2 generation, this T0 generation means suppose right now I am doing a transformation experiment and I am getting a plant from that transformation experiment. So we call it as a T0 generation, and whatever the plants I am getting, we call it as a T1 seeds, or we are getting so T1 seeds I am getting from that. So for T1 generation. So again I have to sow that seeds, and again I have to analyze. So there will be segregation. So some again, because whatever the transgenes we are infecting, this has having only one single copy, and you know that the homologous chromosome will not lack that other copy, right? So it is in heterozygous condition. Okay, it is in heterozygous condition. So whenever we say about the next generation, that is the T1 generation, so this transgene is getting segregated. Okay, and the 3:1 ratio should be obtained if there is a one copy of gene. If we will consider only one copy of gene in T1 generation, and in T2 generation, there we can get a homozygous plants of this transgenics, and this time again there will be no segregation, because whenever we have to say about the variety, we say that it should be homozygous in nature. There should be in a homogeneous population. Okay, so in T2 generation, where we are getting a confirmed transgenic plants, or the homozygous transgenic plants. So T1 generation. So whatever the generation we are using, suppose right now I am doing the transformation experiment or transgenic plants experiment. So that is a T0 generation. Okay, so we have to go to the T2 generation to get a homozygous plants. Then southern hybridization. So southern hybridization is a technique where we can also check the presence of a transgene, and where we can analyze the copy number of transgene. So copy number means, whenever the transgenic plants are being commercialized, they should have only single copy of a transgene. Okay, single copy of transgene. If the transgenic plant is having 2 copies of transgene, it is not being accepted. 3 copies not being accepted. So transgenic crop should have only 1 copy of transgene. And how this copy number is getting confirmed? Is getting confirmed by the technique southern hybridization. Okay, southern hybridization means DNA hybridization. There is one more technique to confirm the presence of transgene, that is a thermal asymmetric interlaced PCR. Okay, thermal asymmetric interlaced PCR. Now with this tail PCR, that is a thermal asymmetric interlaced PCR, we can confirm the presence of transgene, and we can also find out on which chromosome transgene is being inserted, on which chromosome it is being present, on at what position of that chromosome, whether it is located, whether it is integrated at 1000 base pair, or whether it is integrated at one line base pair. So at exactly where it has been integrated in the chromosomes, so that can be found out by the technique of tail PCR. So what is the main purpose of that tail PCR? That is thermal asymmetric interlaced PCR, is to find out the site of integration of that transgene in a genome on a chromosome, because whenever we develop a transgenic plant, we should keep in mind that it should not disturb the function of endogenous gene. Should not disturb the function of any endogenous gene. So that tail PCR is a technique where we can find out the site of integration of a transgene. We can find out where that transgene is located, whether it is in genic region or non-genic region, whether it is disturbing any function of any endogenous gene. Okay. So there is one more technique, that is a plasmid rescue technique. It is also being used to find out the site of integration of a transgene. Okay. So there is one more technique, that is a plasmid rescue technique, which can be used to find out the location of a transgene in a genome. Then these are the presence of transgene we can find out. So what we have seen about the PCR. Copy number of that transgene can be checked by southern hybridization, and we are interested in only one copy if we would like to read the transgene at a commercial level. Then we have seen about the tail PCR and plasmid rescue technique. So that can be used to find out the site of integration of transgene, to know the location of a transgene, because that transgene should not disturb the function of any endogenous gene. Then northern hybridization. So northern hybridization is a technique where we can find out, RT-PCR is there, northern hybridization is there. So we can find out whether it is producing mRNA or not. That the RNA is getting produced. So that RNA level confirmation, or whether that gene is expressing or not, that RNA level confirmation can be done by this reverse transcription PCR or hybridization. Okay. So there is a real-time or qPCR. So qPCR is a technique where we can quantify the amount of transgene. How much amount of that mRNA is being produced. Okay. So quantification can be done of that mRNA, or quantification of that can be done by using real-time PCR or qPCR. Then there is a technique, that is a protein. So as I told you, Western blotting is there and ELISA is there. Enzyme means immunosorbent assay. So these are the technique, Western blotting as well as ELISA, where we can analyze the gene expression at protein level. So where we can come to know whether gene is forming a protein, whether gene is synthesizing a protein, because if it has to synthesize a protein, means first it has to synthesize the mRNA. That can be confirmed by Nelson hybridization or real-time PCR or RT-PCR. You know that real-time PCR or RT-PCR technique is most commonly used whenever we are quantifying the COVID coronavirus, right? Because the coronavirus is made up of RNA as a genetic material, and that RNA we are measuring, right? So it's measurement, quantification. Then if you have to confirm the transgenic expression at protein level, then we are using the ELISA technique. Okay. That is enzyme linked immunosorbent assay. So Western blotting technique, which is also being used. If you would like to see the presence of that protein, okay, we would like to see the presence of. So here we have to use some monoclonal antibody, and we have to hybridize that particular protein with this monoclonal antibody, and develop against that particular antigen. So the Western blotting and ELISA, these are the techniques where we can use the where we can see the gene expression at protein level. So these are the various expression. These are the various techniques which can be used for the confirmation of the transgene. So there we can confirm the amount and quantify the transgenes. So you can unmute yourself and you can ask the questions right now. Okay. So whatever we have seen now, I'll give some time to. So whatever we have seen, there are different methods of gene transfer in plants. There are different methods of the gene confirmation. Okay. So agrobacterial tumefacient mediated gene transfer is the most commonly used method of gene transfer, because it forms a single copy transgene. It means there is a single copy of transgene, and we are interested in only single copy transgene. And the copy number can be analyzed by southern hybridization. Copy number can be analyzed by tail PCR. Site of integration can be analyzed by tail PCR, plasmid rescue technique, where we can come to know the location of that transgene. So please unmute yourself and you can ask the questions, or you can type it in chat box. Please unmute yourself. Please unmute yourself and you can ask the question. So PCR is the technique. So where we can use the total genomic DNA isolated from the transgenic plants, And in PCR, we are using PCR technique, which has been discovered by Carey. There we are using the tag DNA polymerase. Because this DNA polymerase, tag DNA polymerase, which has the property, which can polymerize at 72 degrees Celsius temperature. You know that generally enzymes are having optimum temperature, that is, they are working at optimum temperature, 37 degrees Celsius temperature. But this tag polymerase has a property that it works at 72 degrees Celsius temperature. Means that DNA polymerase have the optimum temperature, that is 72 degrees Celsius, and where that DNA is getting polymerized. So somebody has asked, there is a one good question from Rutu. So what is the difference between PCR and RT-PCR? So you see, our first difference I will tell you. Polymerase chain reaction. RT-PCR means reverse transcriptase PCR and PCR. So PCR is generally, it is the technology. Okay, generally it is a gene specific PCR. We can call it as PCR. So polymerase chain reaction, that PCR is a technique where we can amplify gene or DNA. Okay, particular segment of a DNA, right? Particular segment of DNA we can amplify, means we can produce many copies of that particular genomic fragment or transgene or any genomic region, okay, by using some complementary base pairs like oligonucleotides, primers, right? So, and in case of RT-PCR, that is reverse transcriptase PCR, there we are quantifying the expressions. There we are seeing whether gene is expressing or not. There we can see how much RNA is there, okay? How much amount of RNA is there. In this way, we can quantify that RNA, right? If whenever I say that RNA, so whenever I say that RNA means it is the expression analysis, that gene expression analysis can be done by using RT-PCR. And in case of PCR, only structural presence of gene is there or not, whether that transgene is present in the genome or not. That is only the physical presence. But in case of RT-PCR, we can come to know whether that, if there is a presence, it's okay, but whether that gene is there or not, whether that gene is forming RNA or not. So that quantification can be done, whether that transgene is expressing that particular mRNA or not. So that can be done by using RT-PCR. So by using RT-PCR, RNA can be analyzed in a specific way. And if that RNA is getting converted to the cDNA by using reverse transcriptase, then normal PCR also you can use. Okay, that is gene specific PCR. Any other question you would like to ask? Is the transgene incorporated into both homologous chromosomes at the same time? Very good question. See what I have said you. Suppose right now I am performing a transgenic experiment. Okay, right now I am performing a transgenic experiment. Or if right now I am performing a development of transgene. So what I am doing, suppose right now I am following the agrobacterium mediated gene transfer method. So what I would like, I am co-cultivating that agrobacterium to the plant cell. And whenever that transgene is getting incorporated into a chromosome or into the nucleus, so only one copy is getting integrated anywhere in the genome. Okay, anywhere in the genome. So that is a random integration. Okay, anywhere in the DNA that particular transgene is getting integrated. So whenever this first T0 plants are there, we call that as a T generation. So there is only one copy of that transgene. Okay, means it is not having its allele. There is a lack of that allele. So we know that if there is a lack of that allele, we call it as a hemizygous condition. Okay, so only one allele is there. Because it's only one, that gene has been incorporated. Okay, it doesn't have its complementary allele to the homologous chromosome. So in that situation, we call it as a hemizygous situation. Okay, or hemizygous condition. And whenever you know that hemizygous condition, suppose it is a gene and that particular allele is lacking. So you know that when it gets cell, then it forms a T1 generation. So there will be segregation. That means some plants will have capital A, some plants will have capital A, it will not have allele. So 3:1 generation of transgene and non-transgene. And in T2 generation, we are getting a homozygous lines where you can find that transgenes on the both homologous chromosomes. Okay, so very good question has been asked by someone Reddy. So wherever we generate the transgenic, it is in hemizygous state. So that you should remember. and T2 generation where we are getting a homozygous transgenic lines. So that's where we are interested, because whenever we talk about the release of variety, that should be homozygous in nature. There should be no segregation in the further generation. So that homozygosity can be studied by southern hybridization, that can be studied by quantitative real-time PCR. Because that gives the copy number determination, southern hybridization, where there is a single copy of incorporated or double. We are interested in single copy function with this homologous allele. Okay. Any other question? Please don't hesitate. You can unmute yourself. There should be an interaction. Okay. So, is there any information of loops in pachytene stage for transgene in heterozygous state? Is there any formation of loops in pachytene stage or for transgene? See, in heterozygous state. See, I actually what I have to say I didn't get it, but pachytene is the state where the crossing over happens. Okay. So whenever the crossing over happens, means there is this, that means there is an exchange of genetic segment between the homologous chromosomes, right? And the patches stage means the transgene is getting also segregated in initial generation. Okay. So there is an exchange of but the most important thing, whenever it crossing over happens, the transgenes never, once the transgene is integrated, it never changes its site of integration. It never changes its site of integration. Site of integration will remain the same. Okay. So whenever we think about the commercialization of transgene, so whatever the events has been commercialized, whatever the transgenic crops are accrued commercially, so that first their copy number has been analyzed. So whether it should have a single copy transgene. Its site of integration is also known, and we call that as the event. So in case of BT brinjal, I told you that one event has been commercialized. So in case of BT cotton, MON 731, the event has been commercialized. We call it as the Volgard 1 and Volgard 2. So we know the prime gene location where it is present. So it is the fixed location, that site of integration, and that actually gives the rights to the developers. That gives the right to the patent, because if I have developed such events and I know the site of integration of that. Any other questions you could like to ask? Please have an interaction, please. Actually, I would like to take some more, maybe in next week. I'm covering so many topics. That time also, I will remind all these things. Balaram sir know that I have some other assignments today. So you should be interactive, okay? So you should ask the question. So many questions you can ask on the slides. We have seen about the most of the major breakthroughs, milestones in the field of genetic engineering, in the field of biotechnology, Nobel awardees, their contribution. Okay. So whatever the co-vaccines you are right now, you are that is vaccines you are taking, it's the product of recombinant DNA technology, genetic engineering. Okay. Like insulin, interferons. So these are the recombinant vaccines. Okay. Sir, which books do you prefer for all these topics? See, if you have to clear your concepts, if you would like to clear your concept, BD Singh is good. BD Singh, textbook of biotechnology given by BD Singh is good. Kalyani Publishers. And if you would like to go for any objective type of material, okay, that is, if you would like to do for it for, if you would like to focus for the majorly competitive study, then you can think about the instant notes of molecular biology, instant notes of molecular biology, instant notes of this one, recombinant DNA technology. Then you can also have the Yes Chan publications, the textbook of biotechnology, okay, given by Jain scientists, Jain and Jain. So textbook of biotechnology given by Yes Chan. So for objective type of study you would like to do, and instant notes you can refer. But it should be updated, okay? It should be updated actually what's going on. So any other questions you would like to, you can type it in chat box, you can unmute yourself. A textbook of biotechnology by Eastern Publications. So that is good. Textbook of that was molecular biology, Eastern Notes of Molecular Biology is good. If you have to go concept wise, then BD Singh is good. Book Kalyani Publications.
So you can ask
Any
the
other
question.
questions you would like to ask? Sorry, sir. Actually, I'm sorry because I have to deal with some assignments. Actually, I something more and more.
Sir, your voice is break, sir.
Most of the methods of the deals are.
Sir, your voice
Just check
is
it
break.
right now.
Sir, your voice is break.
Can you check right now?
Hello.
Sir, check it now.
Yes, yes, yes.
Is it okay, sir?
Yes, yes, yes.
Fine.
Yes, yes.
Actually, so we have seen, sir, actually gene transfer techniques and their confirmation and the various historical backgrounds. Maybe
So you can ask the question.
sir.
Sir.
You can. We have discussion.
I kindly request to participant if you have any doubt, query, question.
See, anyway, I will give you a quiz in the form of Google, in the Google form, and you will have the answers there only once you get solved it. So, right now, if you have any questions, you can ask it. You should have a common discussion platform. Okay, it should be a peer-wise discussion. Otherwise, how I will come to know that whether you are getting the right things. So you should have every slides is questionable. I can ask the 100 questions from one slide, you know. So if you have no questions, sir, I think you can the next lecture also we can have some more topics.
Okay, sir. Okay. So participant, there is no question from participant side.
I will give other 5 minutes, sir. I will wait, or 2 minutes. I will give. They
Yes,
can
yes,
have.
yes.
They can unmute themselves. They can unmute themselves.
Most of the people from the biotechnology background present in this meeting also.
See, there is a lot of scientists have contributed so many things. They are Nobel awardee, and I have given the major breakthroughs in the field of science. So there you should remember the concept wise, the Gregor Mendel, then the Hershey and Chase, Johnson, then Kary-Mullis. So many scientists are there. Hershey and Chase experiment. So everything you should remember. Paul Berg, who has created the first recombinant DNA molecule. So which is the technique like for gene expression? So many things can come. Actually, the question should come in your mind. Then only you can ask.
Suvarna madam, present. I think Suvarna Poddar madam.
Sir, I think sir, Bharat sir.
Sir.
Hello.
Sir,
there is no need of vote of thanks. Actually, I'm having other assignments, so maybe because we'll have other lectures also, no?
Okay.
So at the end you can do it, sir.
Okay, no
because
issue.
we are meeting again, so that please, sir.
Yes, no issue, sir. So if there is no question, so I think I should conclude this meeting. So the next time, sir, will deliver the lecture on other different topics. So at that time you can prepare your question. So for the clear your doubt in next lecture.
Okay, thank you, sir.
Sir will give the questionnaires to you. So solve it properly and give the answer properly.
Yeah, give me some 2 to 3 days time, sir. I will provide all the question banks.
Yes. Okay, sir.
Okay. Thank you, sir.
And some give some list of books. So which book we will refer.
Right. Everything I will do, sir.
So
Everything
that
I
is
will do.
very important. So accordingly, they will purchase the book and
Yes,
prepare for the NET IRS examination, sir.
Right, sir. Right, sir. Okay. Thank you, sir.
Yes, sir. Yes, sir. And I will provide your number also and
Yes,
email
sure,
ID
sir.
also.
Sure.
So
Anytime. Right.
individually, they
Okay.
may communicate to you. Those people who are interested.
Right.
Interest is very important. Otherwise, nobody will
Yes.
contact you. We are
Okay, sir.
this training program for participants means those who are preparing for the NET IRS examination. So that's why we are organizing this type of the training program. This is not a one-way program. This is two-way. Your response also important from the participant side. So okay, I think we should stop here.
Okay, sir. Thank you, sir.
Okay, sir. Thank you. Thank you very much.
Thank you.
Okay, sir. So participant, you should join the meeting today's 4 p.m. again. So I will share the link in the group also, and another scientist will deliver the lecture on biochemistry. Okay, so the scientist from the ICAR New Delhi, so he will deliver the lecture. So accordingly, you should join the meeting. This is very important for you peoples. So 4 p.m. Meeting will start, but before meeting starts, you can join the meeting before 15 minutes. Okay.